Well, I am extremely honored to be the last to have a chance to thank Dr. Howard, the Howard Foundation, and the organizers, John and Jane and Neve, for the invitation to be here at this wonderful meeting, and for all the careful, thoughtful planning and how well we've been taken care of and housed and fed, and it's been really lovely. So thank you so much. How do I get to my talk?
Just go forward.
Oh, just the next.
Yeah, yeah. Next again. Yeah, yeah.
Okay. There we are. I have no conflicts of interest to declare, except some of the funding for some of the work I'll be describing was from DSM and Pfizer. And I thought it would be appropriate at the end of this meeting to sort of go beyond macula and go beyond carotenoids, as other people have already done so wonderfully here, and talk about the interactions of xanthophylls with omega-3 fatty acids, and also some more recent work that we're doing on brain imaging and organization that I'll talk about at the end. So we've talked here about a number of dietary factors that can lower the risk of AMD. Of course, the ARIDS 1 and 2 trials, and although omega-3 fatty acids came up a bit empty in the ARIDS 2, there certainly are a lot of reasons to think they may be involved in perhaps the earlier stages of AMD. And I'm also going to talk a little bit about high-fat diets and high cholesterol and a general Western kind of unhealthy diet, which is something that Stuart mentioned yesterday. So there are many mechanisms of action of omega-3 fatty acids that are very plausibly involved in the whole pathophysiology of AMD, including alteration of membrane properties, especially in outer segment membranes, but also throughout neural membranes. Is this on? Yeah. Optimizing of retinal function, modulating ion currents, and of course DHA, as this extremely polyunsaturated fatty acid, 6 double bonds, is very vulnerable to oxidation, and yet appears to have all of these beneficial effects, including reducing oxidative stress, being anti-inflammatory, anti-angiogenic, anti-apoptotic, and neuroprotective, in part through some of its special metabolites, NPD1 and resolvins and other compounds. And as Toss so beautifully summarized, there are many of these protective effects that are shared with lutein. Particularly, he did a beautiful job of summarizing the anti-inflammatory effects. So there's certainly many potential mechanisms for synergy or additive effects. One study I want to highlight is one that Liz talked about yesterday, her study with Max, that's one of the first and few to look at interactions of supplementation with DHA and with lutein, where they found increases in macular pigment with DHA alone, even without lutein supplementation, in the central, particularly in central fovea, and then the combination produced the sort of best profile of increase in macular pigment optical density. So DHA facilitated the accumulation of lutein in the blood and macular pigment. And as Liz described yesterday, also the combined supplement group also showed the best improvements in a number of measures of cognitive function, including memory, learning, and executive function. So I'll be talking the rest of the talk about our monkey model of, actually 2 models of AMD. I think this audience all knows that higher primates are the only animals that have a macula and a fovea that share all the characteristics of the human macula. And that includes the fact that they develop early to intermediate stages of AMD. So as you can see here, Drusen are very common in macaque monkeys of several species, Rhesus cynomolgus and Japanese, all having been documented. And in Rhesus in particularly, these Drusen develop in an age-related manner. They're highly prevalent, and they share at least two of the key risk factors for human AMD, polymorphisms in HTRA1 and ARMS2. So this shows the prevalence of Drusen in our colony of monkeys at Oregon National Primate Research Center. And this is based on fundus photographs of about 1,000 monkeys across the age range. And see the prevalence is very high of all Drusen. That includes small, hard Drusen. But if you look at what we call moderate severe, which is sort of about corresponding to AREDS grade 2.5 and 3, it's up to half of all monkeys in the older age ranges. And as far as age conversion, it's about a 3-to-1 conversion. So a 25-year-old monkey is like a 75-year-old human. And this is just an example of progression over age in one particular animal. Now, we have looked at immunohistochemistry of Rhesus Drusen, and this is something Toss also showed some beautiful slides for humans, and showing that that Drusen in humans express a lot of these markers of complement activity, inflammation, and immune response, and so do Drusen in Rhesus monkeys. They also show the same sort of morphology shown here. And this is a triple label in particular for this MAC attack complex, C5b9, as well as ApoE and vitronectin. And a few other examples here of expression of amyloid beta, amyloid complex, a number of other C5, ApoE. And so for the rest of the talk, I'm going to talk about our animals that have been on semi-purified diets that contain no carotenoids. What I'll be doing, there's been many talks here about supplementation with xanthophylls, supplementation with omega-3 fatty acids. And I'm going to be talking about the inverse, which we can do in monkeys, but wouldn't do experimentally in people, where we deprive them of these molecules. So the animals I'm going to be talking about have been fed these diets from birth, and in fact, their mothers during pregnancy were fed similar diets that contain all essential nutrients, but no carotenoids. So these animals have no macular pigment, and it's been confirmed biochemically by Liz that they have no xanthophylls in their tissues. And within these, they're broken down into 3 groups in terms of their omega-3 fatty acid dietary composition, one being omega-3 deficient, fed a very low level of alpha-linolenic acid, the second a relatively high level of alpha-linolenic acid, and the third fed DHA, EPA, and arachidonic acid in a way that mimics the composition of human and monkey milk. And I could say these studies started when we were looking at developmental effects on the retina and brain, and we're now studying effects on aging, since some of these animals are now 18 to 20 years old. We found that the deficient diet reduced the level of DHA in the retina by 80%, and the same is true for areas of cortex in the brain. As I mentioned, first of all, these animals have no macular pigment, so this is a normal monkey, one with on these diets. And in a study that we did several years ago with Ivan Lung and Max Snodderly, they did very careful morphology of the RPE layer and showed that in normal animals there's a steep peak of RPE cell density in the center of the fovea, but in the xanthophyll-free animals there's a dip in that central 0.5 to 1 millimeter of the retina. Animals on xanthophyll diets develop drusen at very early ages and with much higher prevalence, as shown here, than animals fed a normal diet containing lutein and zeaxanthin. In this study we did also with Max and Liz, we found that there was an increased vulnerability to coherent blue light damage in the fovea. This is at half a millimeter eccentricity in the animals that were xanthophylls free. We also found that in the animals that had the combined deficiency with omega-3 fatty acids, there was an increased blue light damage out in the parafovea at 1.5 millimeters eccentricity. So there were interactions of the two nutrients in terms of blue light damage. We also found increases in lipofuscin accumulation in these animals. In those with adequate, in the ALA group, there was a small increase in lipofuscin, in particular at this 1 to 2 millimeter eccentricity. In those with the double deficiency, a much greater accumulation of lipofuscin, and again, particularly at about 1 millimeter eccentricity. So as we followed a similar group of these animals out to older ages, we have found a number of the animals with this combined deficiency of xanthophylls and omega-3 fatty acids that have developed more advanced macular disease. And this is something that has not been reported previously in monkeys. One of the limitations of the monkey models of AMD is that they don't develop advanced either geographic atrophy or CNV. But these, and we've had 3 animals now that develop this more advanced degeneration, which looks not like classical geographic atrophy, but perhaps like pattern dystrophy. And one of the things that's really interesting about it is in that every case, it's in an arc in the superior retina at 1 millimeter eccentricity. So that corresponds with the area of increased lipofuscin accumulation, and also corresponds with the locus of the increase in blue light damage in the omega-3 deficient animals. And so we're continuing to follow these as they progress, and this shows progression of this syndrome over 5 years with these little maps. Maps down here showing increase over subsequent years of the area that's affected by this type of degeneration, and then that's plotted here, the area affected. We are sort of expecting that this is going to create a ring, but it's particularly in the superior retina, and we really don't know why superior would be more susceptible than inferior in this case. So to summarize these studies, we found that monkeys without macular pigment deficient in xanthophylls show an early appearance of drusen, an abnormal dip in the fovea and RPE cell density, increased lipofuscin accumulation, and increased foveal blue light damage. Those with the added omega-3 deficiency show a further increase in lipofuscin accumulation, increased parafoveal blue light damage, impaired rod sensitivity and recovery, which is not something that I was going to discuss here, although after this discussion about rod dark adaptation, I'm sorry I left that out. And they also show progression to a more advanced form of macular disease. I will just mention that what we find in these animals in terms of rod dark adaptation, actually, this is the work of Brett Jeffrey in my lab, who developed an ERG method for looking at macular, or at least central 30-degree dark adaptation, specifically of rod dark adaptation. And and found that that is impaired in the animals with Drusen. So if you look across the entire retina, there's not an effect on dark adaptation, but macularly there is, and that corresponds with what's found in human patients. And I think we think that that provides a more sensitive measure for use in animal studies. And actually, I want to just go back and sort of respond to one of the questions that Neil had earlier about photopigments. And what our concept of what's going on here is that the function of the RPE is impaired as drusen accumulate and as Bruch's membrane accumulates lipids and forms what Christine Kircher calls the lipid wall. And that therefore the regeneration of photopigment, which involves transfer to the RPE and transfer back to the photoreceptors, that whole process is impaired and slowed down, and that's part of the mechanism for the changes in dark adaptation that are seen. That said, we have also seen reductions in rod maximum amplitude, which are consistent with a loss of rod photoreceptors, which is something that's, of course, seen in aging and with AMD. And now I'm going to talk just briefly about a second model in macaques. These are Japanese macaques, Macaca fuscata, which we have a colony in Oregon. And these animals have they have Drusen, but in a different syndrome. They're pan-retinal. They're early onset, can be identified by even 1 or 2 years of age. Progresses in number and size over the lifespan until there's rather impressive coverage of Drusen throughout the retina. It follows a very clear dominant inheritance pattern. So this is really similar to perhaps the dominant Drusen syndromes that are seen in humans, which are a relatively rare condition, but has close phenotypic similarity to AMD in terms of the form of the Drusen. And I'll show you here that we've also done immunohistochemistry of these Drusen, which also express these key markers of Drusen that are found in human AMD, including here the MAC-ATAC complex, C5b9, C5, beta amyloid, bipronectin, and ApoE. So these are, again, similar in their molecular composition to Drusen in both rhesus and humans. And we've had an opportunity in these animals to look at the effects of what you could call a Western diet. So the classic cheeseburger, French fry, and soda diet. So we have been able to put a number of these animals on a diet that's high fat, 37% of calories, high cholesterol, high in sugar, no lutein or zeaxanthin, and extremely low omega-3 fatty acids. And that's compared with the standard colony diet that's extremely healthy. It contains DHA, fish meal, lots of lutein, zeaxanthin, super low fat. It's like much healthier than anything that humans eat. And so over the first year, in order to be able to compare progression of Drusen on these two diets, we needed to develop a really sensitive way to quantify Drusen, which is actually a remarkably difficult task, and a number of very eminent labs have taken a crack at it. We have an image analysis core headed by Anda Cornea, who took this on, and what she developed was a machine learning algorithm for quantifying Drusen. So this is an example of a fundus photograph. This requires very high resolution fundus photographs. It's contrast enhanced, and then basically the computer is taught how to recognize Drusen versus non-Drusen, run through a few iterations, and then that's used to analyze a whole series for a particular animal over time, photographs that were taken at 3-month intervals. And it uses a suite of machine learning programs that are available free on the web through the University of Waikato in New Zealand called Wika. It's the name of a New Zealand bird. And using this quantification of Drusen, this is what is expressed here is, let me go back a second, the area, the percent of area covered by Drusen within the central millimeter, and in this 1 to 6 millimeter diameter annulus. And although we analyze it by 4 quadrants, I'm going to talk about average for that annulus here. So this is for the fovea, the rate of increase over 12 months in the Western diet group and in the healthy diet group, and similar for the perifovea. And for the fovea, when we correct for initial value for age, gender, we get a significant difference in the rate of progression. It was about an 8-fold difference in the rate of progression over the first year. And although the difference is similar for the perifovea, the variability was too high to see a significant difference, but we'll be following these animals out. I think 1 year is actually a kind of short time to see a difference in a rate of progression. So we'll be following that out. And then for the last part of my talk, I want to go back to our rhesus double deficiency model and talk about the brain and particularly the cortex and its organization, and some new work we're doing with a relatively new MRI method that's called resting state functional connectivity MRI. And this is a functional MRI measure that can be done with sleeping infants, with anesthetized animals, and that looks at the cross-correlation in bold activity. This is blood oxygen level, which is a correlate of neural activity, which has these oscillations over the scale of seconds. And you can cross-correlate those oscillations between different neural areas. And these correlations correspond rather well with connectivity that's measured neurophysiologically or by track tracing or by other kinds of task-induced functional MRI. So it's a very nice method to use these kinds of studies. We have a group at Oregon Health Sciences University that's used this method to look at the developmental trajectory of parameters of connectivity in infants and children, and has found, for example, considerable differences in infants with ADHD and autism. So it appears to be a quite sensitive method. And we used it to look at, first of all, connectivity within the visual pathway, since we know that there are effects of omega-3 deficiency on retinal function. We wanted to follow that through to visual cortex. But we also looked more general at connectivity among all cortical areas and also the larger scale modular organization of the brain. So for the visual pathway, this shows the degree of connectivity between V1, primary visual cortex, and the primary visual relays, the lateral geniculate nucleus and superior colliculus, which you would expect to be very strong. This is obviously a primary visual pathway. And in the animals fed DHA, it was in fact very strong, as shown here. In the animals fed only ALA, it was considerably weaker, and those were omega-3 deficient. It was almost non-significant, which is rather amazing for the primary visual pathway, not to have strong connectivity. And also, when we looked at interhemispheric connectivity between right and left primary visual cortex, V1, that was also reduced in the deficient group versus the DHA-fed group. The same difference did not hold, however, for primary somatosensory cortex, so it was specific to visual cortex. Then when we look more generally at, there are 173 sort of regions of interest or different areas of cortex that have been defined both through imaging and architectonically, and looked at correlations of activity among all of those areas. We got this sort of profile of correlations, strength of connectivity across the cortex. So this is no connectivity and positive, progressively stronger here or negative here. So for both the deficient and the ALA-fed group, there was a rather narrow band of correlations compared to the DHA group, which had much stronger correlations between many more areas and in the positive direction in particular. So that's sort of a much more global measure of function. And then we also looked at modular cortical organization. And this uses graph theory and community detection to group the cortex into areas that are more connected with each other, including widely distributed networks. So this is an example in a typical human brain of these 5 areas that have been defined in terms of systems within the cortex. In particular, these first 3 are very much involved in higher order cognitive function. It's sort of, I find it odd that one of the key networks is called the default network, and what that refers to is that it's active not when people are doing a particular task, but at rest, but it's still, its activity appears to be key for a number of cognitive functions, and also frontal, parietal, and cingulate opercular networks. So those corresponding areas are shown in monkeys in the DHA group here, and you can see that for these animals fed DHA, the organization is remarkably similar to this normal healthy human brain. The arrows sort of show corresponding areas for these 3 networks, but just on a gross level, you can see there's rather broad similarity. And that actually had not been shown before for monkey versus human. And then when we look at effects of different levels of omega-3 fatty acids, this is that same one I just showed you for the DHA group. The ones in the ALA-fed and deficient groups are remarkably different in their cortical organization. They show a rather striking absence or attenuation, particularly of the default and frontoparietal networks, which are critical for cognitive function and perception, and they show more localized networks. And the MRI experts describe this as a sort of more primitive organization of the brain. So we are currently looking at cognitive function in these animals, some higher tasks that tap these networks to see if we can see differences in their cognitive function. So for summary of these results, monkeys with a combined deficiency of both the carotenoids and omega-3 fatty acids had decreased connectivity in the visual pathway, reduced correlated activity across the cortex, and then this altered pattern of large-scale functional organization. Now, we have no idea, because these animals are now 18 to 20 years old, whether these are effects that could have arisen developmentally, whether they're the result of lifelong deficiency, whether they're perhaps happening in relation to aging. And we also don't know the specific role of the lutein and zeaxanthin. We're going to be doing some comparisons of these groups to a reference group of animals on a normal diet high in lutein and zeaxanthin. Obviously, we can't go back and add a fourth group from 18 years ago. But I think really to answer this, we'll need some new studies where we could look developmentally at effects of absence or supplementation with lutein and zeaxanthin to see those effects independently. So with that, I want to thank a large number of collaborators that have been involved in all of these studies. Of course, Liz Johnson and Wolfgang Schalk and Max and Ivan and Felix Barker that are involved in the deficiency and supplementation studies we did previously. I want to highlight the group, the Advanced Imaging Research Center group at OHSU for their work on the functional connectivity MRI, and a number of my collaborators at the KCI Institute and in my lab, and of course funding from a number of agencies. So thank you very much.
So thank you. Yes, question just there.
Man, that's cool. So for a little while, I was doing some work with Jay Kaplan at the Wake Forest colony.
Mm-hmm.
And one thing that happened when they switched all of their monkeys over to the typical American diet, they called it the TAD.
TAD, right.
When we did that, in fact, we were writing up an abstract for experimental biology and kept running into the confound that when we were analyzing the data, these monkeys went through a starvation period. When they offered the TAD to the monkeys, they rejected it flat out and then starved for a little while until finally they got hungry enough to start eating it. And so all of their lipid values and everything went haywire for a little while before they sort of caught on. Did your monkeys like it? Did they eat it? Was it palatable?
Yeah, we eased them onto it.
Uh-huh.
And ours isn't exactly TAD. It's sort of my own version. Similar to the diets that we've used for a number of years, and we had like a 1 or 2 week fade in, after which they've accepted it quite well, and actually, at this 1 year point now, we're starting to get some getting a little heavy. So yeah, we're also going to be looking at lipid parameters and all of those things. But yeah, but no, we didn't really have that. Interesting that they did. Yeah.
They ate it. Huh. It had nothing to do with the
And they went from the standard chow,
You should have given
which
them
I must say I don't find terribly appealing. It's hard to chew. The chow is so high fiber, and it's kind of gritty and crunchy and greasy.
you should have given them a beer first.
Yeah,
They would
right.
have loved it. Yeah.
Right.
My question is a
Yes.
very basic one. You said in your talk that the difference between humans and monkeys is that they don't progress to end-stage AMD. Why do you think that is? Is it just how long they live? Is it genetics? Is it lifestyle? Have you any comments?
Those are all, one of my hypotheses has been that aside from genetics and possibly absolute age rather than relative age, that it might in fact be diet and lifestyle, because the standard, all of the groups that have, the ones we've looked at, and there are a number of other groups that have looked at Drusen in different monkey colonies in Japan and Puerto Rico, et cetera. Those animals are all fed a standard laboratory diet, which is insanely healthy. It's just beyond anything. They're 13% of calories is fat, and super fiber, and no sugar. And so I thought that may be one reason. So that's the hypothesis we're sort of trying to test here, to see if we can push the progression and perhaps push it to more advanced disease. But we really don't know. So probably a combination of all of those.
And did your monkeys develop any other diseases like diabetes or hypertension?
Well, we actually are separately looking. We have at our Primate Center a whole group that's looking at diabetes and obesity models. And the ones that have either become pre-diabetic or diabetic have been on these diet, which is more the actual TAD diet that is mentioned. Some of them are developing non-proliferative diabetic retinopathy symptoms, tortuous vessels, flame hemorrhages. We haven't seen any obvious proliferative disease yet, or leakage, but we're continuing to follow those too. So, we could be inducing a bit of both with the high-fat diet. Yeah, exactly. But one of the things I want to mention is that the animals on those diets, which is designed to induce diabetes, a number of them have also shown remarkably rapid progression of Drusen. So.
Okay.
You showed this RPE, this dip in the RPE cell density. Is this? I have a couple of questions. Is this Lipofuscin related? How does this develop?
I'm sorry?
Is this Lipofuscin related? How does it develop in time? Is this seen in humans too?
Well, unfortunately, we can't look at it over time because it's morphology. So the animals that were in those are the animals from the original lutein and zeaxanthin supplementation study, which were 8 to 16 years old at the time, and pretty much all of the ones that were xanthophyll deficient had this dip. The increase in lipofuscin occurs right sort of at the, there's the peak and then the dip. So the lipofuscin is right about here, not in the very center where there's the loss of RPE cells. So that doesn't quite correspond. But it is really a really sharp peak. It still is nowhere near, of course, proportional to the increase in cone density in the center. I mean, it's still a much lower ratio of RPE to photoreceptors.
How does
in the
that
center.
relate to this lutein-free diet? What's the mechanism?
We wish we knew. I mean, we don't know. I mean, one mechanism is that simply that RPE cells are dying in the fovea, dropping out. But we don't know if there could also be, if they're moving, changing their shape. I mean, we would, yeah, we would love to know what's going on.
But why would they specifically die out in the fovea if there's no lutein present? And what will be the cause? If it's not Lipofuscin driven?
Sorry.
What will be the cause that it's specifically in the fovea that they die out? If it's not Lipofuscin driven?
Well, there may be other effects of blue light besides accumulation of Lipofuscin and the generation of all those toxic byproducts, or that some of those toxic byproducts may not be so local, maybe more general. I mean, we really don't know, but it's very interesting. Yeah.
Okay.
Hi.
On your left.
Okay.
Question.
Oh, sorry.
In terms of the cognitive function, I know you said you're just looking at that now. Just wondering how many time points across the lifespan have you been looking at the development and change in cognition? And do you have any sense at the moment of what kinds of changes you've been seeing?
We did some very early infant measures of cognition, and we found differences in visual attention with the omega-3 deficient infants that actually have been replicated in human infants as a function of DHA supplementation. That's by John Colombo and Susan Carlson in Kansas. But this MRI method is relatively new, and so far this is the first crack we've had at doing it. We are now going to be doing it longitudinally, but now we're into aging, not development, and we're picking up again on the cognitive function. So yeah, we'd take a whole new study to look at the developmental course, which is of course what we'd love to do.
Just a question about when you're looking at Drusen area, Western diet versus standard diet, the baseline values were different.
They were. Yeah, we tried very hard to match the groups, but we didn't have the measurements at baseline.
So could that have affected rate of progression, or?
Yes, and that was factored into the statistical analysis. and yet it is a problem. They were not well matched. It's true. Stuart.
Can you tell us something about the water supply that these animals are drinking? Is it standard? Is it a filtered water supply? Is there iron, copper in the water supply? Copper plumbing? Has that been changing over time?
It's the Portland City water supply, which comes off of Mount Hood, and is very soft water. Actually, we have really nice water supply, so it's not actually hard at all.
Are you checking the iron and copper in the water and zinc?
We haven't specifically. I will go back and check on that. I believe. I mean, we have such level of regulation of everything that I'm sure that has been checked by our vet staff. But yeah,
because there's a theory that these redox reactive metals can influence microRNA changes, upstream passages, which relate to hypoxia-inducible factor and neovascularization. So. It seems important to look at iron and copper. Perhaps.
Exactly. And in terms of inducing more advanced forms of AMD in these monkeys, there are a number of other factors that could be added in to increase progression, and that certainly be one. Yeah. Thanks.
In the first part of your talk, you talk about the monkeys fed with both a xanthophylls-free and DHA-free diet, and then they develop various symptoms of macular degeneration. So how about those monkeys fed with a xanthophylls-free diet, but then supplemented with ALA or DHA respectively? Does it help with alleviated symptoms?
Well, we haven't changed the diets. These animals have been consistently on the same diets throughout their lives. I can say in terms of the Drusen prevalence graphs, I just have combined all of the xanthophylls deficient animals. I didn't break it down by fatty acid group, part because the numbers are too small to look at prevalence, but I can say the ones in the DHA group are kind of the exception there, and that those are the ones that don't develop Drusen early.
I have a follow-up question on that. So, like for those animals fed with a xanthophylls-free diet, so what if in the middle age of life you give them back the carotenoids?
Right, yeah.
What would you predict to those monkeys?
We haven't done that, so we don't know. I mean, we did this one supplementation study, but I think it only went from 6 to 24 months of age, and it was, again, too small numbers, and we didn't see any obvious Drusen regression in those animals, so we really haven't done a study that could answer that.
I'll talk really loudly since I don't have a microphone.
Hi.
I also have a question about the monkeys fed a xanthophylls-free diet from birth. Presumably, at the moment of birth or during their prenatal development, they were receiving some from their mother, or was their mother also xanthophylls-free? And if, assuming that they were born with some level of macular pigment, how long into their development do you think it took them to lose what they had? What mechanisms might be at work there very early in the lifespan for those who and part of my reason for asking that is I think there are probably a lot of American kids, maybe other places in the world, who essentially have xanthophylls-free diets, or at least aren't getting any.
Pretty low.
Yeah. So.
Yeah. Well, in fact, the mothers were on these same on the semi-purified diets throughout pregnancy.
Okay.
So and Paul has also shown, and John had some data too from monkeys, that at birth there are very low, in some cases no detectable macular pigment or biochemically leading to zeaxanthin. And actually we found that too in some infants that we looked at. So there conceivably from mother stores could have been a really low level, but it was never detectable.
Did you track activity levels for any of the monkeys? I'm sure some are much more active than others, and was there any difference if you did in
actually
who had worse degeneration?
Well, we actually, we did a study on that earlier in their lives, up to about 4 to 6 years of age. We did activity monitoring, and the omega-3 deficient animals tend to have a higher activity level and more stereotyped activity. So, you know, one could call it a little bit of more of a, like an ADHD pattern. But we don't have recent data on activity levels. So in terms of relationship to the development of disease, right?
Yeah, just exercise.
Exactly, sure. Sure. I mean, these are cage-living animals, so they're not out running around. They're relatively sedentary.
A question?
Just a quick question. A long-term lutein zeaxanthin-free diet, do you think that could affect the normal metabolic pathways of lutein and zeaxanthin?
One would think it might, but what we found in the supplementation study is that they were still entirely capable of the uptake and accumulation of either L or Z into the retina, into the fovea.
But what to do with them?
Pardon?
But how do they treat these? I mean, how do they metabolize? I know they uptake them after you put them on a deficient diet.
But, you know, are there different metabolic products? I don't know, maybe Liz has some thoughts about that, because she did all the biochemistry.
We really didn't look at that. But as far as Martha said, they still had the ability to take it into the retina. We've looked at the brain and as well, and really don't see anything obviously different.
Yeah, my question is more towards metabolism rather than uptake.
Yeah, we didn't look at that.
They didn't get the values of the controls in the end. It did not reach the actual values of the controls.
Well, by biochemical measurement, they were comparable. In the reflectometry measurements with the lutein supplemented had lutein at higher levels out into the periphery. So it gave us a sort of falsely low value, but by biochemistry, they were comparable.
Okay, so if there are no further questions, thanks a lot once more.